Sure.
So Bhanu, up until this year, was a member of the ENIGMA program as well. He was a postdoc with us. He did his PhD in life sciences at the Advanced Center for Treatment, Research, and Education in Cancer in Mumbai, where he worked on ERK ribosomal kinases. And then he did a postdoc first with our colleague Dror Noy at the Galilee Research Institute, working on iron sulfur protein design. And then he came to our group, to the ENIGMA group, to continue that work. And in January, he started a postdoc with John Peters at Washington State, where he's currently working on characterizing hydrogenase biochemistry. And so today, Bhanu is going to talk to us about selenium oxidoreductases, a promising alternative biochemistry for more reducing worlds. Thanks, Bhanu. Go ahead.
Thanks, Vic, for the introduction. I think it seems to be a very pleasant day to be in New Brunswick. It seems to be 70s, and we are still in 40s, 50 in Pullman in Washington. So yeah, so my name is Bhanu. I was a former postdoc with Vic and Paul, and today I'm going to talk about selenium oxidoreductases, and I'm going to boost them as an alternative biochemistry for more reducing worlds. And the image that we are looking at is of the Titan, which is the largest moon of Saturn. And in the center, we are looking at these are basically liquid hydrocarbon oceans of the Titan. So before I begin my talk, I just want to give a brief introduction to the origins of feroxanes. And the question is, what is a feroxane? It's a very small protein, just about 60 amino acids, slightly bigger than the definition of a peptide. And it's about, it has, as I said, like it's about 60 amino acids and usually have 8 cysteines. And 4 of the cysteines would be in the first half region and the other 4 cysteines would be another half. And I won't call it even as a domain because it's again this 30 amino acids would be too small to call it as a domain. So these 2 half regions are so different from each other. That's why I would call as an asymmetric monomer. Monomer being that it's a single polypeptide chain, and asymmetric meaning that the first half is completely different by sequence with the other half. And that's why I was showing in 2 different colors. The first half would be in red and the other half would be in the blue. And these 2 half regions are joined by this linker in green. So the idea of origins of feroxanes was first proposed by Professor Dayhoff in way back in 60s and it was published in paper Science. And if you look into her paper, she took recently sequenced feroxane, which is probably about 55 amino acids. She just split into 2, just over the loop, which is DSI in this case. And when she overlaid the first half over the second half, she realized that not only the 4 cysteines that are perfectly matching, it's like perfectly conserved. Apart from that, there are a few other amino acids, like in this case, you could see like alanine, say proline, valine. These few amino acids are also conserved along with these cysteines. So that's what she wrote in the line 4. And just taking these conserved residues in the line 4 and wherever it comes, either on the first half or the second half, and by including 4 additional serines, she would build around 30 amino acid sequence, which is like about a half feroxane sequence, and which she calls as the mother or the ancestor for these 2 half regions. So also elsewhere in the paper, she mentions that iron sulfide being the catalyst. Also, she uses the word protoferoxane for the first time. So if we take all these pieces of information and put them together into a flow chart, we would probably come up something like this, which would be for the origins of feroxanes. So these feroxanes, they all started as a very small peptide, say in the order of say 10 or 20 amino acids with 4 cysteines that would be binding 4 and 4 sulfur cluster. And due course of evolution, it has dimerized to become a homodimer. And over the course of the evolution, it has fused to become this homodimers, fused to become a single functional symmetric feroxane monomer. And again, with time, because of divergent evolution, each of these half regions have evolved independently, and that's why that's what led to the asymmetric feroxane monomer, and that's what we see in the modern day living organisms. And these domains are so important that they have even conserved and integrated into more complex enzymes, as we see in extreme right in the hydrogenases here. So Paul and Vic, they have been working on understanding the origins of feroxanes for quite some time. And John, Kim, and Doug, so they worked on this beautiful peptide called ambidoxin, which is only 12 amino acid, has 4 cysteines. It's a heterochiral molecule, and but it can bind 1, 4, and 4 sulfur cluster. And and then Andrew Mutter, who worked on the symmetric feroxanes, and he showed that these are not only functional in vitro but also in vivo, and it was published in PNAS. And I joined after that, and I joined the lab in the group of ENIGMA, and I was asked to see, if this feroxanes are even functional, and if they're functional, what would be sorogomeric state. So I just took the Andrew Mutter's 4 feroxane monomers that he worked on. I just have to split into half regions at the linker region, and I have to see whether if they're functional or not. If they're functional, so whether it could be still a monomer with 4 and 4 sulfur cluster, or it would be like a homodimer and would have 2, 4, and 4 sulfur clusters. So to address that, I started elucidating. I started working on this project, and here is the data. And I'm really sorry for this overcrowded slide, but I just wanted to quickly go through this. These half feroxanes, I would call them as 2AN, 2AC, 2SN, and 2SC. And when we constitute with iron and sulfur salts, it would definitely have the iron sulfur clusters. And this is the UV visible data. And this insert would be when we try to add sodium dithionate, the cluster would be completely reduced, and you would see the saturation of the 400 nanometer peak. And as I said, these are homodimers, but I unfortunately do not have FPLC data to show. But however, I have an EPR data, which, the EPR spectra looks very much like a feroxane, where you have 2, 4, and 4 sulfur clusters that are coupled to each other. And in this bottom left, these are electrochemical titrations, and the midpoint potentials are within the range of say minus 400 millivolts to minus 510 millivolts. Again, these are well within the range of 4 iron, 4 sulfur clusters. So we know that these half feroxanes are functional in vitro. Now the question is whether or not they are functional in vivo. To address that, so we have done, in collaboration with the Rice University collaborators, we have tried to express these half feroxanes genes in the mutant bacteria, which otherwise cannot survive, where the sole source of sulfur is the sulfite ion. So the only way that this mutant bacteria would survive on such a media is to have a functional feroxane gene. And in all these 4 cases, this 4 half feroxanes in all these 4 cases, to a moderate extent, the mutant bacteria had been rescued. It means that these half feroxanes are not only functional in vitro, but also function in vivo. So with this here, I want to conclude that these half feroxanes are evolutionary old, are biologically relevant, and are ancestors to the modern day feroxanes. Now, the question is, because these are so small, so we asked ourselves, can we substitute this sulfur with selenium? And the answer is yes. And that's why we wanted to come up with this selenium oxidoreductases, a promising alternative biochemistry for more reducing worlds. But before I dive into the data, I just wanted to briefly talk about alternative biochemistries. For that, I just wanted to emphasize what is known life on Earth. As you know that the living organism, the non-living organisms on Earth uses carbon compounds, water as a solvent, and the genetic material would be either DNA or RNA. But alternative biochemistries are all speculative because nothing has been proved yet. So would probably use other carbon compounds, maybe use other elements. Say, for example, in this case, I would say propose selenium, and it could be a completely different solvent other than water. So if you look into known already proposed alternative biochemistries that have been proposed over like in the past few decades, the most convincing and the most logically possible would be, in my opinion, would definitely be alternative chirality. So just imagine a world which is just like Earth, and you have the same CHNO, carbon, nitrogen, oxygen, phosphorus, and sulfur, and everything would be the same, but instead of L amino acids, the organisms there would have D amino acids, likewise L sugars. So also there have been several other biochemistries that have been proposed. For example, the notable ones would be the ammonia biochemistry, and ammonia is it's alternative to water, and it's polar and is universally abundant. It's first proposed by Haldane in the Origins of Life. Likewise, silicon biochemistry was proposed by Graham Keynes in its clay hypothesis. Silicon, just like its carbon, is like tetravalent. Likewise, silicon dioxide biochemistry has been proposed. It's alternative to carbon dioxide. And another logistically and logically possible would be alternative nucleic acids, which are otherwise called xenonucleic acids. So again, just take very much like a planet, which is very much like Earth, had again the carbon, nitrogen, oxygen, sulfur, phosphorus, and everything else. But the genetic material, instead of ATGC, might have a different class of nucleotides and nucleosides. So this way, you would have definitely different living organisms living up there. So likewise, there have been other proposals as hydrogen sulfide and hydrogen fluoride, and methane biochemistry has also been proposed. However, selenium has not been emphasized in this front yet. So the question is, can selenium biochemistry be an alternative biochemistry? Before we get into that, we just look into selenium oxidoreductases. Fortunately, they are naturally present on Earth, on many living organisms on Earth itself. So, but selenium in living organisms is mostly bioavailable in the form of selenocysteine, which is the 21st amino acid. And the three-letter representation for the selenocysteine is SEC, and the single letter representation would be U. And it is coded by a UGA codon, which otherwise is a stop codon. So organisms which would have selenocysteine, in those organisms for this UGA codon, there would be a special tRNA that would bring in the selenocysteine at the site of translation. So we look into a few examples. These are some major examples. I don't want to go into its functions, but I just want to read about it, like glutathione peroxidase, selenoprotein P, iodothreonine deiodinase, and selenophosphate synthetase, and also thyrodoxine reductases. So these all enzymes have at least one or more than one selenocysteines in their sequence, primary sequence. So if we try to mutate this selenocysteine with structurally similar sulfur-containing amino acid, which is cysteine, these enzymes would lose their function completely. They become completely non-functional. Apart from these enzymes, there are also certain isoforms of format dehydrogenases as well as hydrogenases. They do have selenocysteine in their primary sequences. It is indeed found in all three forms of life, bacteria, archaea, and eukarya, but not necessarily in all organisms, meaning that there could be some microorganisms which probably does not need selenium at all and can completely live in a media without selenium. So then we wanted to see, we wanted to test whether the small half feroxanes, which are only about 20 amino acids, if we substitute, instead of sulfur, if we would like to add selenium, they reconstitute with iron selenium clusters. And that is what we wanted to test. And when I got this idea, I thought it was quite novel. I was very excited. But when I started looking into the literature, scientists have been working on this for quite a while, and there are papers all the way from 70s and 80s. So this is one example. So a feroxane was, 4 inputs of a cluster was replaced with the selenium clusters and was characterized well. And recently, Esselborn, he was a former ENIGMA member, and the group of Thomas Happe, they took iron-iron hydrogenase, and one of the iron sulfur cluster was replaced with this iron selenium clusters, and the hydrogenase is still functional and happy, and it's good, it's still functional. Likewise, this is the most recent paper from Marcus Rip group. It's just published like 2 months back. So they took a nitrogenase, and one of the iron sulfur cluster of the nitrogenase enzyme has been replaced with the selenium clusters, synthetic selenium clusters. And again, the nitrogen is still active. So again, this suggests that we can functionally replace sulfur with selenium, and we still have the activity of the enzyme. But otherwise, if we try to take a selenoprotein and we try to remove the sulfur group with this, if we try to replace the selenium with the sulfur group, and most of the cases, it happens that the selenoprotein loses its function completely. So this is our data that we have. As I said, these are like very small peptides, so we don't really express in from the bacteria. It's good enough that we can either synthesize in the lab or we actually procure from the companies. So these peptides, so as I said, like I had 4 half feroxanes, but I just wanted to see if the system is working or not. So I selected 2 of the half feroxanes, which I would like AN and the AC series. And this is the mass spec data of the apoprotein, and the mass is very much accurate, close to the theoretically predicted values. And and when we reconstituted with selenium salt, iron and selenium salt, there were clusters, and this is FPLC data to see whether what is oligomeric state. And the FPLC data corresponds to a dimeric nature of these peptides, holopeptides. And in the next slide, I have the data for the UV visible spectra. On the top panel, we have the UV visible spectra. On the top, the green one would be the AN, and the orange one would be the AC. And the solid line is the 4 iron 4 selenium clusters, whereas the broken line is the counterparts with the 4 iron 4 sulfur clusters. You would see, generally, if we see into the literature, if we substitute a feroxane with the selenium clusters, you would always see enhancement of these peaks. And also there's a orthochromic shift that is normally observed. And I do also see the same thing in this half feroxanes. However, these effects are more pronounced in the AN over the AC. In the bottom panel, we have the EPR data for the AN and AC in comparison to 4 iron 4 sulfur cluster. The solid line would be the 4 iron 4 selenium clusters, and the broken lines for the 4 iron 4 selenium clusters. The EPR's data suggests again that we do have 2 4 iron 4 selenium clusters that are coupled to each other. But if you compare the data with this 4 iron 4 sulfur clusters, you would see a drift of towards more lower magnetic field. And also you would see the broadening of the peaks, and it is expected because of the larger size of the selenium ion. And again, in this case also, I would see the drift is more evident and pronounced in the 2AN over the 2AC. And then we have done the electrochemistry to see, to calculate the midpoint potentials. And the orange would be the AC, and the green one would be AN. And the squares represent the 4 iron 4 sulfur species, whereas the circles represents the 4 iron 4 selenium. So if you look into the AN series, for when we have the 4 iron 4 sulfur clusters, the midpoint potential is about minus 467 millivolts. But when I substitute with the selenium clusters, the midpoint potential was lowered to as much as 53 millivolts further, and the midpoint potential is about minus 520 millivolts. On the other side, there is not much difference observed, but still it is a significant minus 24 millivolts from minus 401 to minus 425 the moment we substitute the 4 iron 4 sulfur with the 4 seleniums. So my interpretation with this is because we have not seen much spectroscopic changes either in terms of UV and EPR, and it's probably that could be the reason that is why I'm seeing lower change in this AC. So at this point of time, I would like to conclude that when we try to substitute this 4 half feroxanes with the selenium clusters, we do see a lowering of the redox potential. So with this, we would like to say that these half feroxanes might have an evolutionary advantage when you have a world which is more reducing in nature. So we also wanted to, so for that reason, we're also collaborating with our Theme 3 collaborators like Jiwa. He was a former postdoc with ENIGMA. He's now a faculty at the University of University of Science and Technology of China. So although I'm not an expert in this field, but the take-home messages from his studies were, it's true that selenium is less abundant than sulfur is, and it's also true that selenium has lower solubility. But however, the selenium would have greater stability under more reducing, and also more at lower pH conditions. So so that is why this selenium oxidoreductases would definitely have a greater evolutionary advantage in a more reducing world, and might probably incorporated or maybe incorporated in a life if it really exists. So it's only a matter of time, and future orbital and lander missions might definitely address this hypothesis more clearly. And that's all I have. And thank you all for your attention. And I want to thank Paul and Vic, because this was not supposed to be my main project, but they trusted me that this might work, and they gave me all the freedom to work on this project, although this was not my main project. But and I'm very thankful to Vic and Paul for letting me do all these things. And I'm also very thankful to Alexei for the EPR and Jiwa for the geochemistry data that he's working on. And we are writing a manuscript on this with him. And thank you again, once again, everyone. Thank you.
Questions for Bhanu?
Stupid question. Can you do it halfway? Can you just use the selenium salt and have it be a 4 iron, 2 sulfur, 2 selenium?
Yes, probably we could do that, but I have not done that because it's very difficult, because sometimes we cannot control the stoichiometry. It could be possible that I might end up with 1 sulfur and 3 seleniums. Likewise, I would end up with 3 seleniums and 1 sulfur, also like 2 sulfurs and 2 selenium. But in the time I had, it would be really good to see the EPR and Redox chemistry properties, but it's very difficult to control the stoichiometry, because when you're doing it in, when you're trying to reconstitute, you may have like multiple species also, right? When I add this, so it's very difficult to have a pure.
I hadn't thought it all the way through. That is correct. You would just make a mess.
Yeah.
And you don't want to do that.
Yeah, there's a possibility. All possibilities are there. 1 sulfur and 3 seleniums, and 3 sulfurs and 1 selenium. Yeah, it's a good thing to try, definitely.
Michael.
Hi, it's Mike Wong from Carnegie. Thank you for this really interesting work. You motivated it with Titan, so help me out here. So on Titan, we don't see any sulfur, and selenium's cosmic abundance is orders of magnitude less than sulfurs. So would we expect there to be selenium, very much selenium available on the surface of Titan?
Well, I'm not the right person to answer this question, but well, as Jehovah would have been the right person, but well, if it's all about, sometimes you may have a microenvironments where you may have a little higher proportion of selenium. So it might be possible, or it may not be possible. So, but definitely not on Earth, but some other reducing worlds, maybe still sulfur is dominant, but there could be still some fraction of that particular world, that microenvironment where you may have selenium rich part. Yeah.
Okay, got it. Thank you.
I'm a biochemist, so, but I'm just getting into geochemistry. Yeah, hopefully. Yeah. Thanks for the question.